Welcome to Speaking of Mol Bio, a podcast series from Thermo Fisher Scientific about molecular biology and its trending applications in life sciences. I'm Steve Lewis, and today we welcome a pair of co-authors from the University of North Carolina at Chapel Hill. Alejandro Gomez is a current PhD student in genetics and molecular biology. Jess McAfee recently completed her PhD from the same lab and began a new position at the National Institute of Environmental Health Sciences. Alejandro and Jess have each followed their fascination with CRISPR into amazing early career journeys. We discuss the molecular biology tools behind their work, including massively parallel reporter assays and CRISPRi, and how they use these tools to explore pleiotropic variants tied to psychiatric disorders. We hope you enjoy.
Hey, it's so nice to be here.
Yes, thank you for welcoming us.
So, very interesting stuff that you all have been working on, but I want to learn a little bit about how you came to where you are today. So, why don't we start by talking to Jess first about what brought you into this area of research and how did you get to this point in your career?
I think the chaos of life just kind of brought me to the spot. To be totally honest, I started in plant research and genetics. While I was an undergrad at UNC, I applied to every single lab I possibly could to just do something that had something to do with CRISPR work. And one lab took me and they let me do CRISPR work on plants. And so I did that for 2 years. Um, After I graduated, I applied to a bunch of jobs for a tech position, and I got something in plant research at NC State. Yeah, and then I moved to Japan for a year, and then I came back and I applied to a bunch of jobs, and Heijong ended up hiring me as a lab manager and technician. So very much outside of my plant research, I was really interested in the research, but I really did not know anything about it, to be honest. So a lot of that was learning about, what all these terms like GWAS and EQTLs mean and human genetics and genomics. It's very different from plant genetics. So I was, getting the tea on that. And I really, really liked it. I like really enjoyed it. I found a lot of meaning in looking into something that meant a lot to me, like mental health disorders and things like that. So that's kind of how I ended up where I was.
I love it. And Alejandro, how about you?
Yeah, so I also kind of had a little bit of a crazy, like, I don't know how I ended up here kind of situation. Like, just it basically, like, I started my undergrad in St. Peter's University, that's a small university in New Jersey. And originally, like, I came from high school thinking I just want to do something with CRISPR because at the time that I was graduating, this big paper from Jennifer Doudna with the CRISPR technology being put out was around the same time that I was starting my undergrad. And so I was like, oh, I really want to do something with research with gene editing. I want to be at the forefront of that research. And unfortunately, my university actually didn't do much research work in general, just hands-on. And actually, I started working in a lab that was related to chemistry. So my thesis project for my undergrad was actually related to more like organic chemistry and how we could harness water splitting reactions to depollute waters. And then after that, I applied to grad school because I knew I wanted to continue doing research work. And so my advisor told me, hey, you can do a PhD, and that will be something that I think you would find appealing. And so I applied to multiple universities. That was around the time COVID happened. So it was like not possible to get into any universities with all the shutdowns that happened. So I took a year and I worked in industry after that, working in like manufacturing and things like that, relating to, there was like a couple of jobs. One with was more quality control based, and another one was like related to working with placental derived materials. And then after that, I got into UNC. And so I came to UNC really excited because I knew that there was a lot more kind of work with CRISPR research. And so with Heijung's lab, was actually another student that was part of Heijung's lab that she's like, Heijung is actually interested in starting some CRISPR work soon. So if you're interested in that, you should probably join now because it's an excellent time since she's kind of branching off into that area. And I'm like, oh, perfect. Like, I'm gonna rotate through her lab and I'm gonna see what the project's about. And since then, that's kind of been like the big thing, where I took over a lot of the CRISPR work that Heijon was just starting to do in stem cell derived neurons. So that was a pretty cool thing.
I think a wonderful place to start, since you both shared this, is to ask what was it about CRISPR that drew you into science to the point that you wanted to study it and both pursue PhDs in gene editing?
I think it was just such a new technology with so much potential that I instantly just thought that, like, what are the limits to this? Like, what could this be used for? And how can I get my hands in it and use it too and see what we can do with it? I actually didn't do my PhD with a lot of CRISPR work. That was Alejandro and his collaboration. I used a very different technology called MPRA, which I don't know, we'll probably end up talking about.
I think for me, like at that earlier time point, it was like you could do anything with it because it was just starting. And so actually a lot of new technologies have been developed from the original paper. Like the original paper was like just cut. It just cuts the DNA, double stranded break. And it's like, what can you do with that? You could program it, you could guide the CRISPR to cut anywhere in the genome. And I thought that was cool, but now there have been new ways to use CRISPR, many, many CRISPR technologies. And I think originally it was just like you could maybe fix any disease that has some sort of genetic basis. You could have some way to study it or target it. Now we know it's much more complicated than that, and it's not that like perfect of a technology. We're still perfecting it, but I still think even to this day has a lot of potential.
It's a really inspiring thing when a new technology can be so interesting to someone that they want to pursue the exploration with it, especially one that's a bit agnostic in terms of its ultimate targets that it uh, can impact. So, really, really interesting. Now, you all are both published authors, but you also published a paper together. Can you talk about that?
This paper was really looking into these variants associated with many disorders and what they have in common versus variants that are associated with maybe just one disorder. So the variants that are associated with all these disorders come from something called GWAS or genome-wide association studies. Basically, they take a bunch of people with a disorder and they sequence their genome and they compare that to a bunch of people without the disorder, and they find where those alleles differ in those 2 groups. So I took these GWAS variants and I tested them out in human neuroprogenitor cells, HMPs, using an assay called MPRA, or a massively parallel reporter assay. And basically what this whole assay reads out is, does one allele express more than the other allele? These are regulatory regions, so they're affecting the expression of a gene somehow, some gene that we don't know, because they're in the non-coding region of the genome. So I did these assays, and we got these lists of variants that affect gene expression more or less than the other one. And then we did a bunch of computational analysis that was done by solely in our lab, as well as a lot of other people too. And we found, where these pleiotropic variants that are associated with many disorders differ from non-pleiotropic variants, the ones associated with just one disorder.
When Jessica started, the MPRA was doing it in neural progenitor cells, and they were kind of following up on variants that were associated to many psychiatric disorders. And so when she published that paper and they put it in the submission, the reviewers, one of the first things they asked is like, you have to follow this up with a way that you can connect these variants to actual genes, because MPRA gives you a readout where you know if a variant is doing some sort of regulation or not. But because it only tells you about the variant behavior and how that works in the real genome, you still don't have like a connection to a gene. And so my part came in with actually leveraging CRISPR. And so we leveraged CRISPRi specifically, which is a form of CRISPR where wherever you put it, it just inhibits gene expression. It closes up the chromatin around there. And so one of the things we can leverage about that is that if we want to actually know what gene a variant is regulating, we can just put the CRISPR where that variant is in the genome and then see what gene expression around it is decreasing in response. And so my part in the paper was kind of that. So they had kind of like many multiple variants that could have followed up. So we kind of did a proof of concept study. We took like a few variants and then we kind of just targeted them with CRISPR and just saw what gene expression around those variants was changing.
That must have been pretty new at the time, meta genome-wide association, and then just the approach that you took for integrating, I guess, the pleiotropic hypothesis around it is. Did you have a lot of work that you could refer to before you undertook this?
So GWAS have been around for quite some time. And in the past, I don't know, 5-ish years, like MPRAs have been around to try to validate these GWAS variants. Because GWAS has an issue in which it can tell you the general region that these variants are associated with a disorder, but it can't tell you the like specific SNP because everything in that region is inherited together in these big chunks called like LD blocks, linkage distribution blocks. So the MPRA comes in where it can break up these blocks and test them independently. And that has been done for quite some time. And I had done it previously in a different paper. So by the time we got here, I kind of felt more confident and we kind of knew what we were doing a little bit more. I mean, science is always kind of chaotic, where like you something happens and you kind of figure out how to fix it and standardize your protocols and make things work. And oh, I need more sequencing depth and all of that kind of stuff. But yeah, it eventually got done.
Really interesting, and there's a lot of techniques that I'd like to talk to surrounding that. And before I do, I wanted to mention because I had to look it up to refresh my memory. Jess, do you want to explain what pleiotropy is? Just a short definition for those who may not be familiar.
Yeah, so pleiotropy can actually mean a few different things depending on the context. It can mean that multiple genes are working together in this pathway. But in our context, it means one variant is associated with and potentially causing multiple different disorders. For example, variant 1, allele A, might be associated with schizophrenia and bipolar disorder, and that would be a pleiotropic variant.
And there are other conditions that are similarly impacted. And as I looked it up, sickle cell anemia, Marfan syndrome, just basically multi symptom genetic disorders can be caused from just this ability of a single gene to influence multiple phenotypic traits.
Yeah, so we know from the GWAS data that a lot of these disorders share a lot of genetics. So, how are they being shared, but then turn out as something different later in life? Like, if we are sharing a lot of the same genetics, how does one person end up being diagnosed with schizophrenia and another person end up being diagnosed with bipolar disorder? And that's something that we were kind of looking into. Like, do these variants that are shared between multiple disorders have something in common, some underlying pathway that's affecting what happens to these people throughout their development and later in life. And it's that different from the variants, the non-pleiotropic variants that only affect one disorder and not the others, or are only associated with one disorder and not the others.
Do environmental factors impact in any way?
Oh, most certainly. We did not test that in this experiment, but many people have looked into environmental factors for all these different psychiatric disorders, and I'm sure that environment definitely plays a role.
Hey there, Speaking of Mol Bio listeners. If you're fascinated by the power of digital PCR and how it's being used to transform molecular biology, we've got a new podcast you won't want to miss.
That's right. We're Lisa Crawford.
And Jordan Ruggieri.
Together, we're the hosts of Absolute Genius, the podcast exploring digital PCR and how it's used alongside other technologies to support diverse applications and drive innovation.
Whether you're into bioproduction, cell and gene therapy, oncology research, or just love geeking out on the science of absolute quantification, Absolute Genius has something for you.
So after this episode, come join us. You can find Absolute Genius wherever you get your podcasts, like the app you're likely in right now, or you can visit thermofisher.com/absolutegenius to learn more. That's Absolute Genius, where genius is spelled GENEIUS, with no spaces or dashes in the URL.
Absolute Genius, bringing you absolute quantification and absolutely great conversations.
Moving on a bit to talking about the techniques that are used in the lab, we've spoken a bit about leveraging CRISPR, but what other molecular biology techniques are common in this area of research?
Yeah, so for my side of the story with the CRISPR targeting of variants, we usually have to begin by making a library of guides. So we start by making a list of variants that we're interested in targeting, and then we have to design guide RNAs for them for the CRISPRi library. And so we begin by basically designing those guides. We do some QC to make sure they have good efficiency rates and everything. And then after that, we order the oligos in bulk. And then we kind of clone them into a plasmid backbone that contains basically, it's like a standard crop-seq backbone that we use that also has RFP fluorescence so that we can later leverage it to select for cells that took the guide plasmids. And so we basically put the guides into the plasmid. We do the cloning, a bacterial transformation and selection. And after that, we do a maxi prep to get basically representation of the whole guide library. And then after that, we can basically package it into lentivirus. After that, we deliver into cells. And basically, the only thing left over from there is, in my case, we do single cell RNA-seq, as I mentioned. So we actually have a core at UNC that takes care of that for us. And they basically do library prep for the gene expression libraries for all the cells that we targeted, or the neurons in my case. And then the only thing left over is doing the guide RNA enrichment, which just requires some steps of RT, because the core gives us the cDNA to process from the expression of the cells. And then from there, we process also guide RNA enrichment, which is basically some extra PCR steps to enrich the sequence of the guide RNAs in the transcriptome of the cells so that we can get a better detection rate of what cells had which guides. After that, we just send it to sequencing after those libraries are finalized.
Impressive, just the breadth of techniques that you use. How about you, Jess?
Yeah, so MPRA is along the same line. We, get these GWAS variants. We have to order them from some kind of company as oligos. I do PCR to add on these barcodes to them. I use restriction digestion to add them into a plasmid. I'll put those in bacteria and then extract the plasmids out. I'll do restriction enzyme digestion and ligation again to put it in a minimal promoter and GFP or luciferase or whatever is going in there. And then again back into the bacteria and then back out. And from there, I would put them into cells and then I would extract the RNA and DNA from the cells using kits of some kind. And then I would do RT-PCR on the RNA and then PCR on the DNA. And then another round of PCR to add on our sequencing adapters for Illumina sequencing. And then we'd sequence that. So I did so many PCRs, I would be filling up 50 mil conical tubes of my master mixes. So yeah, we've done it all.
How has it been making a transition to a lab that's run by the federal government compared to academia?
It's very different in that the lab sizes are quite smaller. So you're interacting with the PI and maybe one other person on a daily basis. Where in Haejung's lab, it was very, very large at certain times where we'd have 20 people and we'd all be really chatty and running around everywhere. A bit chaotic sometimes, but in a really fun way. Yeah, the NIH has a lot of resources for us to use, a lot of core facilities that are not as busy because there's less people. So it's quite easy and nice to use the core facilities. Yeah, a lot of structural things are quite different. Like in academia, the PI is busy with writing grants, securing funding for their research and for their students. At the NIH, they have allotted money based on how long they've been there. So they're not really writing grants as much. They're more involved in doing the research and training and writing papers and analyzing data. Though they still have review processes they go through, it's just in a very different way.
Just that description alone, I think, paints a really nice picture of just how different it is, right, from writing grants. What keeps you motivated, Alejandro?
Oh, that's a good question. I think that, if you love what you do, even if the uncertainty of the situation can become quite large. I mean, I always see the big reason why I think doing the PhD, especially from the beginning, seems so appealing to me is because I felt like it is work, but it's like it's work I enjoy a lot, and it feels almost like I'm playing and I'm just thinking and doing things and tinkering, and so I think that keeps it exciting, and especially when I'm doing new things, which you get a lot of opportunity to do new things in, especially academic labs. I think that's the biggest motivation to me that, there can be this uncertainty in the background, but you come into the lab every day and you can do something cool and cutting edge and novel. I think that keeps me excited and keeps me going. So I think that's my take.
Yeah, like Alejandro, I do love playing around and being in lab and doing stuff. I think all sciences have this like inherent hope to help someone somehow, some way. And through the knowledge that they gain with their research. Like for me, I hope that the research I do ends up helping someone with a psychiatric disorder, live a life that is better to them. And whatever way that is, I'll just be a rung on the ladder, of knowledge that just gets built upon and built upon until that happens. And I think that's what keeps me going.
Adding to the body of human knowledge, I think, is a really inspirational and motivating factor for sure. Thank you both so much for your time today. I always close out the podcast by asking a couple of questions. And so we'll start the first one off with Jess. What have been the keys to your success?
My key to success has to be great mentorship and working with really good people. I couldn't have asked for a better lab in my PhD. Haejung is amazing, and the people that I worked with, like Alejandro and Sol and Jisak and everyone else in the lab, are also really amazing people to work with. Yeah, that's my key.
Jess said it best. I think I always remember when I was starting the PhD, one of the things that there's two big things they emphasize when you're choosing a lab from your rotation. They say you need to look at whether the mentor works for you, the mentorship style, whether the lab environment works for you, you get along with other people, and then it just whether you like the science. And I think the science is easy to like, once you're getting into these programs, you know what you're gonna do, what you like, but it can be harder to find, I think, a good mentor and a good lab environment. So I think that that's also similar to Jess. I mean, we come from the same lab, so it's hard not to think of it that way. But Haejung is, I think, an excellent mentor. She's very well connected, which I think is also a huge plus, right? I think for us, that ability to have these, through Haejung connections for after grad school to move on to other positions, just from people she knows, is a huge thing that I think can help with our success. And then also, from the day-to-day experiments, like I know John is very good at like talking to people and seeing what's going on in the field, and then going back to me and being like, hey, I heard of this kind of technique and you've been struggling with this, so maybe we can try that instead, and that can boost you on your way, right? So I think a good mentor is like that. It's like looking for ways to make your life easier in the lab, and then like she said, I think the people in the lab are also a huge plus. I think if there is, kind of animosity or difficulty in relationship with your lab mates that can make everything harder or more stressful. So I think having a healthy lab environment is also a huge bonus.
For anybody who might be listening and maybe much earlier in their scientific careers, what advice would you give them?
My biggest advice is don't give up, even if you encounter initial obstacles. Like, I know for me, when I was just applying to grad school for the first time, this kind of thing with COVID happened. And actually, that was a difficulty even before graduation and going on to apply to grad school. I like couldn't do some internship opportunities that kind of got missed. And with the world environment and the way things change all the time, you never know, right? So you can run into these obstacles where something can be really out of your control. And it may seem, and I know to me, it felt initially at the time like, oh, like maybe it's not meant for me. Like I'm having these issues because, it's just not meant to be. But I think that, and especially for science, it's really good to be persistent and it pays off. And I think that in general is a good thing, not just for science, but for life. So I think for me that's been the biggest thing. It's like be persistent and eventually you will get the results you want.
Love it. Advice for younger scientists, Jess?
I echo Alejandro's. Don't give up. With your experiments, things aren't going to work all the time. You have to figure it out. It's going to be frustrating. Like, find people to help you. Reach out to mentors, to other people in the lab. Ask for help when you can. Use your resources. I think also life is really chaotic, and you just gotta do your best with what you got sometimes and make it work. And that's totally fine too. I also applied to grad school 3 times in a row before I got in, 3 years in a row. It'd be like that. Yeah, just stick with it and just do your best, and whatever happens, happens. And you can just choose to be happy with what you got.
Just a marvelous attitude and really a great story from both of you about resilience and perseverance all the way through a lot of difficulty in your professional experience so far. So, thank you for sharing. We have a little bit of time, so a bonus question. We'll start with Jess. What technology are you most excited about right now that may have an impact over the next 10 years?
Don't judge me, but AI is gonna make such a difference. One person can only do so much. I say this all the time. One person can only have so much in their brain, can always read so many papers, but if we use AI, you can combine so much data together. It can code so much faster than one person. It can like analyze data. It can explain things to you in a way that is just so more efficient. Obviously, with all the check boxes, you need to make sure that it's correct information. You make sure the code is doing what it tells you it's doing. You have to check your work. You can't just trust it blindly. But I think it's really going to revolutionize genetics in general because there's so much data out there that isn't combined together yet into one thing. And when that happens, and it's currently happening, it's going to be really revolutionary. And I think it's going to be very beneficial to us with, all the caveats that come with AI usage as well.
I'm always excited about new CRISPR technologies whenever I see in terms of the science side of things, or of what we do. I'm always excited about seeing new CRISPR technologies. I feel like there's always new things that I see, so I'm always very excited to keep up with that. And any new methods, really, I feel like I'm a little bit of a methods person that I like when there's something new or like a new way that even the current stuff we've been doing, like with MPRA or CRISPR, like somebody thinks of it and gives it a new twist. And so that can also be really exciting to see because you're kind of, it reminds you that you're at the forefront of the field and you see these changing happening. I think that can be very exciting when you look into the future.
I love both of your outlook. Alejandro, Jess, thank you so much for your time today. We really appreciate it on the podcast.
Yeah, thank you.
Thank you.
That was Alejandro Gomez, a PhD student in genetics and molecular biology at UNC Chapel Hill, and Dr. Jess McAfee, a scientist at the National Institute of Environmental Health Sciences. A quick note that the views, thoughts, and opinions shared by Jess are solely her own and do not reflect the views or positions of the National Institutes of Health. Join us next time for more fascinating discussion about the wide world of molecular biology. Until then, cheers and good science. Speaking of Mol Bio is produced by Matt Ferris, Sarah Briganti, and Matthew Stock.